welcome to week 3 of HCDE's research seminar series. So, yeah, yay! All right, so today it's my pleasure to introduce Dr. Shana Hirsch. Shana is a research assistant professor here in HCDE. She just started in that role in May officially, but before that she was a research scientist and a postdoc, so she's no stranger to HCDE, but she's newly in this role as a research faculty. Shana is also co-director of the Pacific Marine Energy Center, or PMEC. And so, really long-standing and nice center, sort of thinking around, alternative energy sources using marine energy. And so, but Shana is a sociologist by training, and so she's sort of navigating this whole world of cool technologies, but from the perspective of sociologists. So, with no further ado, I'm going to hand it over to Shana.
Great. Thank you, Julie. Thanks for coming, everybody. As Julie said, my name's Shana Hirsch. I'm going to be talking today about human-centered design for biodiversity monitoring in the Global South, which is something it's a project that I've been working on for the past year. So I'm really excited to present this body of work. A little bit of an outline of what I'm going to talk about today. I'm going to talk about something called eDNA. So there's going to be a little bit of a lesson in what eDNA is. I've been working with the eDNA Collaborative, so I'm going to tell you a bit about them. And then I'm going to talk to you about the problem that I've been investigating, which is around capacity and standards and capacity to meet those standards, and the studies that I've done to look into that. So eDNA, what's going on here? How did I get here? So I actually am trained as a sociologist of sustainability and natural resources. My degree was in water resources, and I wrote this book, which was published in 2020, and it's all about ecological restoration and monitoring in the Columbia River Basin. So back in 2014, when I was researching this book, I came across this really cool new technology that people were starting to use called eDNA. And I thought, well, that's really interesting. And I put it in my book, and I thought about it and put it on the back burner in my mind. And then I moved on, and I started working on a lot of renewable energy work and marine energy work. And then last year, some folks at the eDNA Collaborative reached out to me, and they were like, do you know about eDNA? And I was like, well, yes, I do. And they were like, do you know somebody who might want to go and find out about people using the technology in the Global South? You might have someone in HCDE who might want to do that. And I was like, well, yes, I would like to do that, please. Thank you. And so that's how I got involved in the eDNA Collaborative's work. So the eDNA Collaborative is located over in SMEA, the School of Marine and Environmental Affairs, where the director is Ryan Kelly. There's also the chief scientist, Eiley Allen, Ada Nip, who's our research scientist, and a project manager. They have a facility over in some of the shared laboratories, and they also have a center over in Wallace Hall. So then I was added in as what they're calling the networks and standards person, which is really code for the social scientist. So the mission of the eDNA Collaborative, it's funded by the Hewlett-Packard Foundation, and the mission is to disseminate and reinforce science that brings environmental DNA analysis out of the lab and into routine practice. So that's what I'm going to be talking about here today. And these are the ways that they do that. So they're providing direct assistance to emerging researchers around the world in the form of grants and microgrants. They're building networks of practitioners and making the information more accessible. And they're also conducting original research to make eDNA tools cheaper and more reliable. So there's this broad swath of science going on over there, all the way from the spectrum of me, from a social scientist, to some really groundbreaking molecular biology work as well. And these numbers are a little old, but you can see that as of, I think this was 6 months ago, we gave out 150 microgrants in 47 countries, and we had 13 visiting scholars from 9 countries, and we have a lot of interaction with folks from around the world. So what is this eDNA thing that I've been talking about? I'm going to tell you about it. And I'm really excited about it, so I'm hopefully not going to talk too long about it. So most of these pictures are from folks who have worked with the eDNA Collaborative. They have got micro grants, and they have sent in photos of themselves doing science. So that's what most of the photos are in here. So eDNA, why are we talking about eDNA? So the technology has really rapidly advanced, and that means that it's become more reliable, the costs have rapidly decreased, and it's become more available. And that has happened really since this 2014 when I first ran into it. Most people are just now starting to realize that this is a technology that they can use. And so it's a sampling method, and as a sampling method, it's non-invasive. And that means you don't have to harm anything in the environment. You don't have to electrocute fish or gather bugs or do anything. It can be done really quickly, and it can be done at a large scale. And there's a lot of opportunity to integrate eDNA methods into routine monitoring of the environment that's happening already. There are also, yes? I'm going to get to it. Thank you. Don't worry. We're getting there. I'm telling you why it's important. So it can be used for invasive and endangered species detection. It can be used for biodiversity monitoring, impact assessment, bioassessment, fishery stock assessments. It can detect disease and pathogens, harmful algal monitoring, and it's expanding into other areas where there's air DNA, there's iDNA, which is insect DNA, there's eRNA, and there's also ancient DNA, which is DNA that is thousands of years old. So here we go. Here's a nice little cartoon which shows you some of the places where eDNA can be gathered. It can be used for trying to understand impacts from industry, which relates to the marine energy work that I've been doing. People are talking about using it for offshore wind farm monitoring. It can be used to detect invasive species, detect species on land, and biomonitoring. So it's DNA that is shed from an organism. So it's basically, there's DNA floating around everywhere in this room right now that's coming off of our bodies. It's on everything. If you go in a swimming pool, it's in the swimming pool. Maybe the swimming pool's chlorinated, so it might go away quickly. But in a pond, there's DNA from all of the organisms that have swum through it, or in the ocean. It's usually fragments of DNA. It's not whole DNA, but there can also be cellular DNA from protozoa or bacteria as well. So hopefully that answers your question. It's basically, if you think forensic science and people going into a crime scene and finding DNA, that's eDNA. If you are taking a tissue sample off of an animal, that is technically not eDNA because it's coming off of an organism, although we kind of blur that line a bit as well. But it's DNA in the environment. So here you can see it comes off of fish and mucus, waste, decaying matter, and then there's some dynamics that interact with it, including the shedding rate and the metabolic activity and the way that things decompose. So there's all of these complex dynamics that are interacting on the DNA as well. So how do you collect it and sample it and analyze it? This is like a very cartoon sketch, but the simplest possible way to describe it is that you sample, you filter, you extract, and you analyze. So sampling, I'm talking about aquatic DNA in most of this, just because that's most of the eDNA science that's occurring right now, and it's also most of our work at the eDNA Collaborative is either fresh or marine eDNA. So there's a lot of sampling tools. People are using drones. People are using backpacks to draw up water. It can come from trawl nets. People are sampling at depth. There's a lot of different ways to sample water. You would run that water through a filter. And again, there's a lot of different filter types. This one on the left is used a lot for citizen science projects. It's just a syringe. with a filter in that little circle part. There's self-preserving filters in the center there, and then you can see on the far right, that's what a filter would look like if you filtered like some pond water or something kind of dirty. But all of that is, not only soil and minerals, but also DNA. Then you extract the DNA. I'm not going to get super into this part, but there's a few different ways to do this, including PCR, which you've all heard of from our COVID experience, and metabarcoding. And these are some of it can go all the way from, these lab technologies to something really simple that you can do on a boat. And this is Aiden, who works with us at the eDNA Collaborative on a boat there, sampling coral reef fishes. And for the analysis, you decide what you do by the type of information that you want to get. So you can either you can quantify the DNA and quantify the biomass that way, or you can identify a single species or multiple species. And then now genomics and metagenomics would bring in, like, say, adaptive factors that are working on different organisms. So I'm not going to get too much into the weeds on that, but hopefully that gives you an idea of what eDNA is. And here's a look at some of the equipment and how it's becoming faster and smaller and cheaper. So this is a paper that Aiden wrote in 2020 with some colleagues. And right here, you have an eDNA lab. You have on the left, you need a laptop. You have on the right, this is what's called a bento lab, which is a small extraction and PCR machine. And in the center, you have a sequencer, which is a nanopore, Oxford nanopore sequencer. So that can be a field lab now for people to do eDNA studies, which is a huge difference from what it has been even a decade or two ago, when you would need a huge laboratory infrastructure to be able to do this work. And so here you can see some more of this equipment that's coming on the market now. And on the right, somebody who's doing biomonitoring in the jungle. So this has really opened up the possibilities for people around the world, and for people that don't have access to sequencers and labs. For example, on the continent of Africa, there are only a few sequencers, and most of those are for humans. So now people who are working in biodiversity have access to sequencers for a couple $1,000s, which is a huge change, and that's only in the past few years. This is the Oxford Nanopore MinION. It's $2,000, and it plugs into your laptop, and it sequences a lot, a lot more than most computers can handle, most laptops can handle. So what have I been researching around this? So there's a problem, and that is that we've got all these technologies that are becoming more and more accessible. People are starting to use them around the world, and people are getting very excited about being able to take these up and use them in their work. They might be natural resource managers, they might be conservation workers, they might be scientists or citizen scientists. And there are no standards for eDNA work. There are forensic standards for working with humans, but there's no standards for eDNA. So there's a lot of mistrust around what is acceptable. Say if somebody finds an endangered species in a pond where they're going to build a Walmart, that might have real legal and development implications. And so people want to know, like, what are the standards? How has this been tested? And this is something that will really help and support the field to grow. So I'm thinking broadly in terms of standards here, though, and this is kind of standard protocols, methods, data and metadata, the materials, the technology, and certifying labs and personnel. And there are no global standards right now. We just got the first person in the US connected with the International eDNA Standards Task Force and the ISO. So they're starting to work right now on international standards. But these standards are mostly drawing on ones that have been written in Canada, Japan, and Europe. And so there's a question around, what if these standards are made to be the international standards, and then folks in other parts of the world might not be able to meet these standards, and they're completely left out of being able to use this technology. And so these are the questions that I was asked to look into. Could people around the world adhere to these standards? And the simple answer is no. And if not, what should the standards be? So that's what I'm looking into. So the real issue that we see is that, eDNA analysis is really changing the way that we can understand and measure the environment, and that is a given. If you want to talk about it, I have so many fun examples of what eDNA can be used to do. And right now, this work really requires a certain amount of expertise and equipment to generate useful information, but that is changing. And it is true also that researchers without easy access to resources are at a disadvantage. And so we're trying to lessen that disadvantage. You have here an image that somebody sent to us of them doing their eDNA research in sub-Saharan Africa. And you can see this is a very different lab than we might have here at the University of Washington. So I've been conducting I'll talk about 3 studies that I've been working on. The first one was a collaborative manuscript. This was a really fun exercise. I highly recommend it. I engaged with researchers around the world, and I asked them what their perspectives were on international standards. I engaged with 18 people. These are all the co-authors. And I really wanted to target areas in the global south and also remote and indigenous communities. And so I reached out to people and I asked them if they would like to be involved in this project. And what I did was I interviewed them all, and then I coded the interviews, and then I drafted a paper, and then I sent it around to everyone. And I said, is this, is your voice in here? Is this what you see as the issues and the problems? This is what came out of this. And we had this really fun iteration over a few months where, in Google Docs, people worked on the paper together. There were some differences that were kind of argued out and settled in the comments. If we couldn't settle them, we got on some calls and we worked things out. And it was a really fun way to write a paper. Part of my reason for doing this is I've done a lot of papers where I interview like 20 people and then write a paper. But I was like, I want these people to be co-authors. I want this to be a real participatory paper. And so that was so much fun. We all learned a ton about each other, and it was people all over the world. The one caveat was that it really required a lot of explanation about qualitative social science to be published in the journal Metabarcoding and Metagenomics. So I did a lot of translation work pushing this through the review process, explaining how qualitative research works and why I was not including any numbers whatsoever in this study because there was, no way to statistically analyze a group of 18 people from a global population. So it was a lot of fun, and I was really excited to see it actually come out recently. So it is published now. Here are some of the results from it. So people did not have a universal definition of standards, but a lot of people use ad hoc standards. So things that they adopt and they say, okay, this is going to work for me. I'll use this as a standard. A lot of people would adapt national standards from Canada or from Japan, places that have national standards, and adapt it to their situation. A lot of people use standards for data repositories. So a lot of folks are very used to standardizing their data. But other than that, there's not a lot available to people. And so there were also a lot of benefits people saw from standards. Reproducibility, of course, ability to collaborate around the world, being able to guide their research, increasing the credibility of the method, and using a common nomenclature, and also having accessible and clear pipelines. Yes? Well, we have a lot of different kinds. So going back to here, like there could be a protocol could be a standard. So say like this is how you extract DNA from fresh water, and it will walk your way through, and it'll say, draw 1 liter of water, use 3 replicates, these kinds of things. And that's really, there's not even that basic protocol standard, but also there are standards for data and how that is reported. Standards in technology could be like, this thermometer is ANSI, is certified by a standards organization, so you can use it. A lab can get certified as a forensic lab, but there's no certification for eDNA labs. So, does that help? Okay. Yeah, feel free to ask questions. It's great. Okay. There were also a lot of concerns about international standards. And I'll say too, like these varied across the world and in different situations. And so one of the challenges of writing this paper too was using language where people felt included, but also where they didn't feel like lumped into one big category of the global South, which is obviously very diverse. So some of the concerns were around access and capacity. And of course, this has to do with resources, equipment, materials, but also sterilization standards for a lab. And a lot of those could be just completely unreachable. A lot of times it takes a long time for a needed chemical to be shipped to a country. It could be ruined by the time it gets there because it hasn't been kept cold. It could be very expensive. A lot of times things are like 3 or 4 times more expensive than they are here because, we have a supplier, 20 minutes away where we can get a courier to run over with an assay, really quickly, and it's all frozen all the way. But if you're shipping something to a remote island or even to Hawaii, it can be very difficult to get materials there. Technical concerns were, should we have a standard that applies both to the jungle laboratory or a laboratory on a boat and to a laboratory at a research institution? Should there be a one-size-fits-all or different kinds of standards? People were also concerned the technology is still in flux. Is this going to mean that we're locked in? And I think that a lot of people kind of were also educated through this, that that's not always the case with standards, that standards are also very flexible. And then there's a lot of concerns around data as well. And the legacy of genetic colonialism is very real. Many people don't want genes to be taken out of a country, shipped far away, especially if it's from an endangered creature or a culturally important one. And a lot of the international standards require geolocation of the sample, and many people do not want people to know where an endangered species might be or an important cultural food resource. So what came out of this, and there's this code here will link to the article if you'd like to read it. What came out of this is that we need to create adaptable standards, potentially flexible standards that include a need to have versus a nice to have level. Developing regional capacity was something that was very important to people. So that would be developing in situ, locally driven regional hubs so that people could do all of the work within their own country and they don't have to ship their samples, from the Congo to to Germany, for instance, and then have to deal with all of the black box of where that genetic material is going. So sensitizing to data sovereignty is another theme that came out of this, and people really resonated with what are called the CARE principles for indigenous data governance, and so we're pushing for that to be included in international standards. And then finally, we really are saying, let's view the standardization as a global capacity building exercise, as a process, where if we're having an international standard, it should mean that everyone has the capacity to meet it. So let's actually make this about bringing everybody along and up to the standard instead of creating a standard that people can't reach. Okay. So I'm going to quickly go through the next couple of activities so that we have some time for questions. So I've also done, I'm doing 3 interactive workshops in the Global South. So the first one was in Kigali, Rwanda at the Association for Tropical Biology Conservation Conference in July. Then I was in Belém at the International Barcode of Life Conference in September. And the third workshop's going to be in New Zealand at an Southern eDNA Society conference. So one thing I just wanted to point out is that when you go to do a an interactive workshop, you have to be very, very flexible about how that workshop is going to go when you get to the place that you're doing it. I didn't have any idea how many people were going to be there, what kind of resources I was going to have, what the room was going to look like. And you can see how very different the rooms can look for a workshop. So on the left there is the Kigali workshop. You can't see that it was 90 degrees in there. It was a very dark, very loud, distracting environment. In Belém, Brazil, it was very different, very open, but there was a lot of people moving around, and the snack bar is right behind us. So, it can be, you just have to be very flexible when you go out to do this research in the field. But the aims for the workshops, again, were to understand barriers to using eDNA and explore more about how standards might support or hinder the uptake of the eDNA tools. And then the results from these workshops are going to be given to the International eDNA Task Force so that they can take all of this into account. So I did a kind of gallery walk where people could post their thoughts. I wanted to get a lot of different, to include a lot of different ways for people to express their opinions, partly because there are people from all over the world that are speaking all different languages. So I had, a sticky note, gallery walk. I had a discussion group aspect, a facilitated discussion group, and then I had a kind of vote where people could decide on their most important barriers. And so I really wanted to have multiple ways for people to express themselves. And that worked out really well because it gave, different people different voices. Different ways to voice their opinion. And so what I did was I took a kind of simplified workflow for eDNA, and I had on each flip chart, I had a different station with a different stage, and people could walk around and put their negative aspects or things that they were concerned about in red, and then things that they thought would work well in green, and kind of allowed people to express their opinions that way. And I got a ton of data, so it was very, very fruitful. I also went and visited the local labs. So this was a lab in Kigali, Rwanda, and I talked to the people who work there. And they have great equipment, but they don't have anyone that knows how to run it. So I was also able to get perspectives from people who are actually trying to do the science in these places. So finally, I'm working on a bunch of projects now that I just wanted to briefly outline. We're doing a kind of user experience study on a new hand pump that the eDNA Collaborative is thinking might be useful for people to use in the field. It costs $2 instead of a hand pump that costs around 500. So I brought with me hand pumps and handed them out at all of these workshops, and people are getting back to me with their experiences of using them in the field. I'm also starting to work with this company, Bento Lab, which makes this little mini laboratory that you can take with you. It costs about $1,000. And I'm trying to see if there are ways that people in remote places can find them useful, if there are things that maybe they don't have access to that make these difficult to use. So this is kind of a phase that I'm moving into now, is using the Bento Lab. And one of the things I'm really excited to do is to work with citizen scientists and potentially school kids, high school kids, to detect salmon in some of the streams around in our area. So this is something that you could like bring out to the field and do an eDNA analysis. I'm doing a diary study where myself and some other folks are trying to, I'm a restoration ecologist, but I've never done genetic work before. So I'm working with some other restoration ecologists to see if we can train ourselves to find endangered amphibians. So I'm keeping a diary along with these other folks while we do this, and we're hopefully going to be able to figure out where we need to develop some information to give to people if they would like to be able to take up this method. Finally, I am really looking towards the open source community to develop. People are really interested in developing low cost but still high tech solutions for eDNA around the world. And so there's a burgeoning open source community that's building around this. And it really lends itself to community science. So I think that there's a lot of places where human-centered design can have an impact on this field. And so my conclusions, which are going to sound really obvious, but I really sometimes think it's important to say the things that are really obvious, is that there is a lot of need for human-centered design in this space. I mean, I just put a short list of things there that could be useful to do. But it goes on and on. People call us every day at the eDNA Collaborative wanting the skills of human-centered design. And so I think that you can find emerging technologies in really surprising places. I just happened to be attuned to this one because I had heard about it 10 years ago. But it really made me think, what other emerging technologies are out there that we're not even thinking about that could really use our help? And I think one of those areas is environmental sensing and biodiversity, and conservation technology, and also the Global South. And I think that here's another obvious point, but I really think that people don't know that they need human-centered design. That, the people that called me up, I knew them from running into them on the campus. And so they knew about human-centered design and engineering. But most people don't know what human-centered design and engineering is. They just happened to know me and happened to call me up. And they didn't even know that they needed a human-centered designer. So I think that there is a little bit of a burden on us to translate our methods into a language so that people in these diverse contexts can really go like, oh, I know who I need. I need a human-centered designer. And that I think that the methods and perspectives that we have here can really support technology and society in ways that we're not really thinking of. So with that, I think you should check out the eDNA Collaborative website. We have Slack and Twitter, and thank you to the Packard Foundation for funding the eDNA Collaborative. I have time for questions. Thank you.